So what I want to talk about now is actually some of the details of transcription. That is a reminder, making an mRNA copy of DNA so protein can be made. And so that's the goal of this. So we're going from DNA to RNA. So in DNA there are 4 letters. That is A, G, C, and T. In RNA there are 4 letters. A, G, C, and U. So the conversion is pretty simple. DNA is 2 strands. RNA is 1 strand. Right? And this is done by an enzyme called RNA polymerase, or RNA Pol for short. Not a super imaginative name, but very descriptive. It polymerizes RNA. It makes polymers of RNA. And a couple of things. The note here is that only one, or so therefore, only one DNA strand is read, right? Which is the first sort of odd thing, but true. And the other thing to remember is that only the part of the chromosome corresponding to the gene desired is transcribed. That is, again, every cell in your body has all the same genes. You just don't need them all at the same time and in the same place. So any given cell is only making a subset of the possible proteins you care about. So you're only transcribing the genes that you care about. So let me draw a schematic of this and then go into final level of detail. If you have a chromosome, it's 2 DNA strands that are base paired. Right? And if you look at one gene, so the chromosome is a long piece of DNA. And I'm not going to draw them all in. There's a zillions of base pairs, thousands at least. And it extends off in both directions. Okay? At one end of the gene, you have one signal sequence. And it's usually drawn with an arrow. This is the start transcription signal. This is a DNA sequence that is recognized by RNA polymerase. It's called a promoter. And in GeneX, it's TATAA, ATATT. That's its sequence. So let me back up and explain a bunch of pieces here. If the point is to only read part of the chromosome, RNA polymerase is supposed to make a copy of a part of the chromosome. How does it know which part to make a copy of? You got to tell it what to do. Right? And you also have to remember how molecules work. They can't see in the distance. They can't think. They don't have memory. They just bumble around. So what you have to do is you make a sequence in the DNA that RNA polymerase will recognize. How does it recognize it? Hydrogen bonds, ionic bonds, the whole 9 yards. Basically, the RNA polymerase bumbles around the cell. If it bumps into DNA, it goes, what's that sequence? Oh, that's not a promoter. Never mind. Finds another part of the DNA, grabs on it, goes, oh, that's not a promoter. But if it happens to find, like in GeneX, a simulated situation, finds TATAA, goes, oh, that's the right place. I'm going to start making RNA. Sequences in real organisms are typically longer than just 5 nucleotides. But that's the basic principle. There's a sequence of DNA, particular bases, TA, TAA, GCC, AAA, whatever, some particular sequence that RNA polymerase has evolved to recognize. And once it sees that, it grabs on and starts making RNA. And that's how it, and so that's also, so for example, if you were trying to get any RNA, you would, unless you have this sequence in your gene somewhere, you would not get any RNA. And if you had this sequence and you messed it up, you changed it, then you will not get any RNA because you haven't told RNA polymerase where to start. Right? So that's the signal to start. And what happens then is the RNA starts getting made, and the RNA goes along, and it has the 5' end is what corresponds to the promoter, mRNA, and this is one strand. Okay, and you've got the same deal, right? You've got a RNA polymerase. You don't want it to keep going forever. You only want it to read this gene, so you got to tell it when to stop. Again, RNA polymerase doesn't know anything, doesn't have any foresight, doesn't have any smarts. It's going to have to be a DNA sequence. So there'll be if you want it to stop at the end of the gene, at the end of a gene, you have to put a signal to tell RNA polymerase to knock it off already. You've made enough RNA. So there's a sequence in the DNA. The stop transcription signal. It's called a terminator. Okay. And in GeneX, it's the G, 5 Gs over 5 Cs. Okay. In real life, the signal is a bit more complicated than that, usually longer. But GeneX is just for simplified purposes. But it is a particular DNA sequence. When RNA polymerase gets there, it goes, OK, you told me to stop. I'm going to stop making RNA. And when RNA polymerase gets there, that's where it puts the 3' end. That's the last nucleotide it adds, and that's it. OK? All right. Before I talk about particular details, which strand, how you figure all that stuff out, I want to make sure everybody's with me with promoters and terminators and starting and stopping RNA. So that's what you would have seen, would probably have driven you crazy. That green and red block indicated the start and stop transcription signals. And you have, although you don't really need a stop, once you get started, you can make some sort of a strange RNA. But you got to have a start, otherwise you can't make any RNA at all. RNA polymerase will ignore your DNA. Questions about that stuff? Oh, yeah. Are those the only letters that work as a promoter? In GeneX, yes. In real life, it's a longer sequence, and there's some slop allowed. Like, for example, TACAA might also work. Okay? In real life, it's a little more complicated. And like I said, longer sequences typically 30 nucleotides long, but that's just a lot to write. But it is a particular sequence. Yes? So the U's, if you saw U's, they would have been in the RNA sequence. Right. Other questions? Yeah. Do the signals get so do the signals get translated? I would fuss about terminology. Do the signals get transcribed? And the answer is no. The signals are not transcribed. So promoter and terminator, actually that's a good, I should make a note about that. Promoter and the terminator are not transcribed.